Assalamu alaikum warahmatullah. This lecture regarding placenta and fetal membranes. The objectives are to identify and discuss formation of placenta and fetal membranes, and starting with the uterus. The wall of the uterus, as you study in previous lectures regarding female reproductive organs, the uterine wall formed by three layers: endometrium, myometrium, and perimetrium. And here we would want to focus on endometrium. It's a functional layer of uterus where the uh, conceptus implant. So we can identify three parts of endometrial cells which at time of implantation rich and we can found accumulation of lipid and glycogen. So this layer can be differentiated into decidual cells which form the decidua. Also, at the time of implantation, the endometrium rich in vascular and uterine glands that support the conceptus until it forms the placenta. We have here three types of decidua. And the decidua is a functional layer of endometrium and arranged in three regions: decidua basalis, decidua capsularis, and decidua parietalis. Decidua basalis, it's the layer of endometrium that found outside of implantation. Decidua capsularis that cover the area of implantation. Decidua parietalis, the remaining part of endometrium. As we see here, here we have decidua basalis, which is in contact with the chorion, and here we have decidua parietalis and become complete in contact with decidua capsularis. Here, the area of implantation or implantation site, surrounding tissues or surrounding layer is decidua capsularis, and the remaining part is the decidua parietalis. And here is magnification for Chorionicvilli on chorionic sac, and here we have decidua capsularis, and deep there in beneath to the implantation site we have decidua basalis. So this figure showing the yolk sac and embryo with chorionic wall rich in chorionic vessels, and here we have the chorionic villi and chorionic cavity. The yolk sac or umbilical vesicle is early site of blood formation. Also, from there, primordial germ cells formed and then migrate to primitive gonad. Also, it share in formation of primordial gut. And here we have yolk sacs that connect the formed or the formed midgut to the umbilical vesicle, which later, if persist after birth, called ileal diverticulum. Allantois also arise from the yolk sac as a vesicle or diverticulum. In this figure, shown this section of an embryo, and here this is a cloacal membrane, this is the allantois, this is a connecting stalk. That's before the tail fold. And here, after formation of tail fold, this part incorporate to the hindgut, and the cloaca and cloacal membrane incorporate to the future anus. And here we have the allantois. The allantois is formed during the third week as a diverticulum from the caudal wall of the yolk sac into the connecting stalk. At this time, from third to fifth week, the blood formation appear in its wall and its blood vessels form the umbilical arteries and vein. So the intra-embryonic part of the urachus becomes median umbilical ligament after birth. If the allantois remain, its uh, extra embryonic part is called allantoic cyst. Here we have the embryo or embryonic disc. Here we have the yolk sac or umbilical vesicle. And here we have the amniotic cavity, and this is a chorion with villi and connecting stalk right there. And here you can see the formation of blood and blood vessels. And then after folding, the cloacal region incorporates with the anal or anus, and here share in allantois share in formation of urinary bladder. And here we have the hind gut right there. In case of male, so this part incorporate to formation of urinary bladder and surrounding or related ducts of male, which share between reproductive and urinary tract. In case of female, share in formation of urinary bladder. The remnant part of it after birth is covered with peritoneum and form median umbilical ligament. Regarding other membranes, we have amnion and chorion. The amnion, as we mentioned formerly, is formed here as a cavity just in the area of superficial to the epiplastic cells and is lined by amnioplast which originated from epiplast. And here the yolk sac and it's lined by Husser membrane for firstly and the roof formed by hypoplastic cells. And here we have the chorion with chorionic villi which share in formation of placenta. So it forms regarding amnion, it forms fluid-filled membranous amniotic sac surrounding the embryo and fetus. Later on, after fetal folding, the amnion enlarge gradually and obliterate the chorionic cavity as the embryo grow, and so the amnion and chorion become contact to each other. Here is the growing amniotic cavity with fluid, and here we have the chorion, and finally the amnion and chorion become contact together. Regarding the umbilical cord, at fifth week, this embryo shows structure passing through the primitive umbilical ring here, including intestinal loop with allantois and two umbilical arteries and single umbilical vein. And this transverse section shows structure at the level of umbilical ring, including vitelline ducts and vessels. And here we have two umbilical arteries and single umbilical vein and allantois. And here, this is transfers section through the primitive umbilical cord showing intestinal loop and two umbilical arteries and single umbilical vein, and here the allantois. The amniotic fluid, it's come from amnion and maternal blood. Also may contain fetal urine and incorporate with skin of fetus, respiratory tract, and membranes. It's composed of 99% of water and 0.25% protein, including also glucose, vitamins, growth factors, antibodies, antigen, etc. It's circulating or changes about every 3 hours. The functions of amniotic fluids protection from injuries, and also it's maintain fetus at constant temperature, also allow movement of fetus and allow symmetrical growth. Also it has antibacterial activity and enhance pulmonary development. Regarding amniocentesis, it's done by needle guided by ultrasound. Needle just pierce all this flare skin and uterus to reach the amniotic cavity and just for take samples from the amniotic fluid, and this called amniocentesis. And the amniocentesis, we culture the cells of fetal organ for chromosomal studies at karyotyping or enzyme measurements or DNA analysis. Also, we can estimate alpha fetoprotein levels because it's indicated in high levels, neural tube and body wall defects. In low levels, it may indicate Down syndrome. So, in case of large amount of amniotic fluid, which is called polyhydramines, it's measure more than 2000 ml, it may indicate microanencephaly or multiple pregnancy or diabetes mellitus and esophageal atresia. In case of low amount of this fluid or amniotic fluid, it's called here when the levels less than 400 ml, it's called oligohydramnios. It may indicate renal agenesis or urinary tract obstruction or placental insufficiency. Also, amniotic band syndrome or sequence. This may cause deformities in many areas by make a mechanical pressure on subjective organs. And this figure may show amniotic bands here in the area of the face and here in the arm. It may lead to mild or severe deformation. Regarding the chorion, the chorionic wall is formed by trophoblastic layer, and the trophoblastic layer lined by somatopluric mesoderm, as we mentioned in previous lecture. This chorion also associated with villi or Chorionicvilli. As the second week, the primary villi appear on the surface, and later, after third week of development, we can see the secondary and tertiary villi. Already we discussed the different villi regarding primary, secondary, and tertiary. And here, this is the chorion and its parts. The smooth part surrounding the fetus or embryo is called chorionic leaf, while the part incorporated with the placenta called chorionic frondosum or chorion frondosum. Here is the chorionic villi within chorionic frondosum, and here is the chorionic leaf or smooth chorion. This is decidua parietalis, and here decidua capsularis degenerated, and here in light color is decidua basalis. And here, after contact of amnion and chorion, we have amniochorionic membrane right there. Sometimes we need to test the chorionicvilli by operation called Chorionicvilli Sampling, which done by searing for aspiration. Also, we can get it by bare vaginal route or bare abdominal route. Two operation guided by ultrasound. For just memorize, the chorion has a leaf and frondosum, two parts, which are embryonic or fetal origin. The decidua, which related to the endometrium, we have decidua basalis, decidua capsularis, and decidua parietalis. The placenta is composed of two parts, maternal part or maternal side and fetal side. Maternal side, chorion frondosum, and fetal side, chorion frondosum, and maternal side, decidua basalis. Together, they form the placenta. So the placenta is a discoid shaped organ about 20 centimeter in diameter and 2 centimeter in thickness. Its main functions are protection, nutrition, respiration, excretion, and hormone production for the uh, fetus. The maternal surface is rough, which filled by cotyledons, and the fetal surface is smooth and attached umbilical cord. Here is the maternal surface of the placenta with cotyledons, is called cotyledons, which are elevation separated by grooves. This is the fetal surface, smooth surface, and here the umbilical cord attachment. This is the vessels there, or chorionic vessels appear there. And here is the full term placenta, the fetal side, and here we have maternal side. This is the decidua basalis. When we remove the decidua basalis, we can easily see the cotyledons right there. It's around 18 in number. So here we have the amnion and we have the chorionic plate. And here, those are the chorionic vessels. Here, you can see the placenta, the fetal side, the maternal side. Those are the cotyledons here. The decidua basalis, rich in spiral endometrial arteries and veins. And here we have the villi or tertiary villi and intervillous spaces which rich in maternal blood. So the maternal blood here surrounding the villi right there, and from which here The spiral arteries finally enter the cotyledons for uh, nourishment the embryo, while the venous return from umbilical artery reach this area and again move to venous or maternal venous side. So the exchange occur in that area of villi and surrounding sinusoid filled with maternal blood. And here is histological figure showing the villi with fetal capillaries inside the villi. Between them we have intervillous spaces which contain maternal blood. The functions of placenta: exchange of gases, nutrients, electrolytes, vitamins, hormones. Also, transmission of maternal or maternal uh, absorbance, also for hormone production. Also, we have the placental barrier. So the fetal blood and maternal blood do not mix directly. Also, the area where RH incompatibility appear. So we can raise these factors different between maternal and fetal without interruption. So this diagram showing the placenta, and here we have the fetal side and maternal side. We have umbilical veins which carry nutrient to the embryo, and umbilical arteries which carry the waste product to the maternal side. And here we have the fetal capillary, and here we have the maternal side. So the veins directly from fetal side go to the maternal venous system toward the lungs and kidneys, while the blood or arterial blood from maternal side which carry oxygen and nutrient, water, carbohydrate, amino acid, lipid, electrolyte, hormones, vitamins, ions, trace element, and other substance also may carry drugs, poisons, or other teratogenic factors, viruses, toxoplasma. They reach the embryo or fetus by endometrial arteries. Regarding membranes in twins, if we have dizygotic twins, that means the twinning happen from implantation of two different zygotes. That means we have two different oocytes at the same time fertilized and become zygote, and the two implanted in the uterus, so they have a different amnion and different chorion, different placenta and umbilical cord. You have two separate placentae. In case of dizygotic, two zygotes implanted just near together, the chorionic sac may be fused and the placenta may be fused also. In case of monozygotic twins, that means the twins occur in from single zygote, that cell mass inner cell mass divided into two regions. So their fate are two. They have two amnion, but we have one placenta and single chorion. Again, we have single placenta and single chorion, but two amniotic sac. Here we have anastomosis of placental vessels and arteriovenous communication between circulatory system of twins. Also, in case of zygote with two morulus, the morula in early cleavage stage or in early development stage at cleavage becomes two morulae. The two morulus then make two different plastocysts. So it can be implanted either in single place or different places. If they become away from other, they have every one of them their own placenta, own chorion, and own amnion. If they implant near together, it may have two placenta but fused placenta and fused chorion, but two different amnion. In case of twins that come from uh, division or separation of embryonic disc, we have single placenta, single chorion, and single amnion. Only we have two umbilical cords, and this may lead to conjoined twins or parasitic twins because the twins are in same amniotic sac. So conjoined twins may appear in cases of uh, separation or division of embryonic disc. Regarding placental insertion, if the placenta inserted and embedded for almost 3 layers or almost the wall of the uterus, it's called placenta precreta. If it reaches the myometrium, it's called placenta accreta. If it's inserted at the region of internal os, it's called placenta previa, and in this case, it's close the cervical canal or childbirth canal. For any question regarding this lecture, just ask your question on Telegram. I'm going to answer you. Thank you and good luck.